So yeah, I'm here representing Lattice Biologics. We're a Scottsdale firm, and we are known for producing, we're a certified tissue bank, and we produce tissue allografts for all sorts of applications, including orthopedic surgical intervention, as well as we have dermis tissue products. We have tendons for sports medicine injuries. And I want to talk a little bit about extracellular matrix-assisted cell viability, because we, as a tissue bank, want to step into the stem cell space and bring a new generation of tissue allografts for improved healing. So here's an example of some of the bone allograft material that we currently have in our product line. Allografts are kind of our business. A lot of these are derived from cadaveric tissues and are used in spinal fusions and orthopedic surgeries. Usually is a void filling, wound healing approach. They're decellularized, and even some of them are demineralized, like the cancellous block here. And I'm going to talk mostly about bone, because it's something that's kind of the first mission of ours, is to improve upon our bone allografts. So maybe it's just wishful thinking, but hopefully sometime in the future, it'll be just as simple to repair a human being as it is to repair a car. And so we envision that we could play a part in this and provide parts that would be superior to existing technologies. The gold standard for, say, bone healing is to use an autograft, which would be tissue from the patient that is taken from a donor site. But there are complications with that, and with tissue morbidity and difficulty with the second site healing. So allografts are the next best answer. Now, we're focused on extracellular matrix because we think that's kind of an approachable technology that we can derive from stem cells that would provide a number of different signals that could accelerate the healing process. So you see all of the cues that a pluripotent stem cell can respond to, or not, in the case of quiescence. But as stem cells have the capacity to self-renew and differentiate, they can also apoptose. And so we would like to use what we know about the extracellular matrix to stave off apoptosis and wake up cells from a quiescent state so that they can both proliferate and differentiate and engraft onto allograft material and become a new standard of care. So the extracellular matrix is rich in all sorts of protein signals, and soluble factors are also present in the milieu, which we don't intend to dissect and identify in any sort of rigorous manner. We just want to use the natural extracellular matrix that's available, but that from stem cells that could aid in the healing process, with an eye on the future of eventually creating the ultimate allograft, which would be autologous, have better regenerative properties, faster surgical recovery. And some of the steps along the way to get there, we would start with our traditional allograft material. Here we have a bicortical dowel allograft, which we would intend to impregnate with native extracellular matrix, growth factors, maybe cells, adult stem cells, which are derived from adipose tissue or bone marrow or something of that nature, that have been grown under an optimized nutrient media composition to encourage the right kinds of signals and to give the best proliferative capacity. So these are the burning questions that are driving our strategy right now to our next generation allograft development. And I include the Phoenix sun here, which I miss after only 2 days. We're in Scottsdale, but it's part of the Phoenix metro area, and I haven't seen snow for 5 years. So I'm already missing the Phoenix sun. I was wearing shorts 2 days ago. So the burning questions driving us right now, one would be, how do we define that culture media that will enhance propagation and promote differentiation from isolated stem cells? How to enrich for stem cells and promote healing in transplanted allografts? And then finally, how do we wake up those stem cells once we've derived them from tissues? So to answer the first burning question, or at least to address it, we're developing technologies in collaboration with the academic lab group at Montana State, who are modeling fluorescent chimeric RNA aptamers. This is kind of work that is based on the Jaffrey Group's technologies, those of you that know about spinach. And the idea here is that these, you can select for RNA can mimic three-dimensional protein-like structures. And you can select for these RNA molecules from a library using CELEX. Now, the process for making a chimeric RNA aptamer is to do this kind of selection process, but in our case, it's going to be two different RNA aptamers that are joined together, one of which has been selected to bind a fluorescent dye, and the best example of that would be the chromophore for GFP. Whereas the other domain has been selected to bind a metabolite. And then those two RNAs are stitched together, and they can be expressed in vitro in induced pluripotent stem cells in order to basically screen for the right ones. And the right ones should give off a fluorescent signal when they've identified and bound the target metabolite. So this is a really powerful technology, we feel, that could help us optimize nutrient composition of media that we would grow stem cells in. And we would look at levels of RNA or levels of metabolites, related metabolites, using different colors of chromophores. So you could have, you could look at the balance of a certain metabolic pathway and determine whether your cells are going to be optimized for transplantation. So that's the idea behind the development of this technology, that we can multiplex and that we can bind metabolites and not necessarily protein. So here's a good example of the applied technology. The balance of glutathione in its reduced state and in its oxidated state, there's GSH being the reduced state, GSSG being the oxidized state. The balance is important for the readiness of cells for transplantation. A high redox balance would indicate a more proliferative, undifferentiated state, and a lower redox balance would be more indicative of a differentiated state. So this is one example of a pair of metabolites that we would analyze using the chimeric RNA aptamers. We would measure the fluorescence output using a multispectral imager or fluorescence microscopy and be able to tweak our nutrient composition to see the effects of certain nutrients that we put in the media on readiness for transplantation. And now back to the ECM, and I guess this addresses the second burning question, which is how do we get those cells to engraft onto the allograft? And so the idea here is to use ECM generated from stem cells. It's a complex milieu shown here with cell surface proteins like integrins and fibronectin, recognizing components of the basement membrane, or the ECM, which provides a number of signals which can direct the stem cells towards migration, proliferation, or differentiation. So here's some of the approaches that you'll find in the literature, and the simplest being to grow cells in a monolayer in tissue culture plastic. Those stem cells then deposit their ECM, and you can then decellularize the tissue culture surface and recycle the ECM that's been deposited. And what we intend to do is actually transfer this onto a scaffold coating There are some folks, some groups that are developing ways of growing stem cells onto scaffolds. We think that's a little beyond us. There's so many issues of perfusion and shear stress and bioreactor conditions. We think that a better approach is to simply transfer the ECM that's been deposited on tissue culture plastic and move it onto the scaffold later. So the main idea is that we want to exploit the ECM for its potential as a reservoir of signaling molecules, and not as concerned with the functions of tissue structure and cell adhesion, although those apply here, because the reservoir of signaling molecules that the ECM provides in fact, can instigate cell proliferation or a homing response, cell migration, or differentiation. Now, in the case of differentiation, let me remind you that our decellularized bone allografts already contain or retain bone morphogenic proteins. So the decellularization process has not stripped it of all of its instructive potential, but differentiation is kind of the endpoint, and we would like to encourage engraftment, vascularization, and attachment to the allograft prior to those cells choosing their fate. And we're encouraged by what we see in the literature. This is a group, DeCaris produced this work at UC Davis, and he and his group optimized the conditions for in the media in which they would test the ability of a decellularized matrix that was laid down by the stem cells in tissue culture flasks for their ability to stimulate osteogenic differentiation. So it appears that normoxia is actually the best for giving that kind of a response, as shown by calcium deposition here. But on the flip side, hypoxia, we believe, is actually an important parameter that you can manipulate, or the oxygen tension, you can manipulate it, and hypoxia would then favor proliferation. So here's some more evidence from the literature. The first one I already talked about. Actually, it's this one right here by DeCaris' group that showed that the alteration of environmental culture conditions during matrix deposition affects the capacity of decellularized matrices to modulate cell fate. The other thing that DeCaris showed, which is really pivotal, is that you can homogenize and transfer these decellularized matrices, and they will retain that same osteogenic differentiation potential comparable to the native matrix that was laid down. Now, there's another group in San Antonio which does some really exciting work. This is Chen's group, and they showed that bone marrow ECM that was laid down actually extends the proliferation potential of stem cells that are laid down on the decellularized matrix. And probably most excitingly, when they compared ECM from a young donor compared to that from an older donor, they found that the young ECM had the ability to restore the functional capacity of those cells, the cells from older subjects, and they would act young again. So if there isn't a better argument for the fountain of youth in a bottle, I think we found it. So this is work that I did back at UC Davis's Center for Healthy Aging, where I was looking at chondrocytes from rat. And as I mentioned before, the ECM is important for adhesion and attachment. There's also, this is kind of to make the point that the ECM is a source of ligands for integrins. And we were looking at fibronectin, full-length fibronectin, against two different alternative domains, the V region, the 3-10 region. We were able to show that when we were measuring global translation, and this is by uptake of radio-labeled methionine, we showed there was a stimulation in cells that were attached to fibronectin in its various domains compared to the same cells that were kept in suspension. So this shows that waking up idea, third burning question. How do you wake up these stem cells, which may become quiescent, especially if you're culturing them in vitro prior to transplantation? How do you wake them up? Well, you may have to have the right collection of ligands that are recognized by their cognate integrins. Now, the chondroprogenitor cells, same, there's a parental cell line for the previous one. The RCJ 3.1 cells, they actually act a little bit differently when exposed to the same ligands. And V and 310 give a partial stimulation, but full-length fibronectin gives an even more robust stimulation of global translation. Now, I don't have the same suspended control here. What I did in this experiment was I laid down polyL-lysine. Now, polyL-lysine will get the cells to attach, but this makes the important distinction that although you get attachment of your MSCs, you may not get the same kind of stimulation of global translation unless you are binding stimulatory or binding integrins that activate stimulatory signal transduction pathways. And so if you look into the literature, you will see that there's a small but growing body of work that discusses the integrin-dependent translational control. And I come from a translational control gene expression background, which is where I did my graduate work at LSU Health Sciences Center in Shreveport under Robert Rhodes. And now here we see that those three different fibronectin ligands, I should say, that were put down actually are recognized by different heterodimers or different integrin pairs. So the 310 region is recognized by alpha-2 beta-1, full-length fibronectin by alpha-5 beta-1, and then the V region, alpha-V beta-3, but they feed into different pathways or similar pathways, ultimately impacting the translational machinery, which gives the go-ahead to start producing protein. And I think looking at the translational side of things could be kind of a key to understanding what would shift cells from a quiescent to a more active regenerative state. And then finally, DeCaris, again, shows that he actually identified alpha-2 beta-1 as the cognate integrins that bind the transferred decellularized matrix that they did the proof of concept in. And however, this was looking more at osteogenesis, which we're also interested in, as well as encouraging proliferation. And he followed up by looking at some of the flux through some of the signal transduction pathways that impact translation. But interestingly, there's a number of encouraging bits in the literature that shows that not only do some of these stem cell derived extracellular matrices encourage proliferation or differentiation, but in this case they can stimulate or prime cells to be more responsive to bone differentiation signals. So this was RT-PCR. They followed the gene expression levels of two different proteins that are involved in bone differentiation, and basically showed that the ECM gave a more robust response. The cells grown on ECM as compared to tissue culture plastic. And so this is kind of our goal, is to tap into a synergistic approach to bone healing. And a former colleague of mine, Nadia Kornieva, also at LSU Health Sciences Center in Shreveport, showed that by engaging alpha-6 beta-4 integrin, the translational control pathway would be specifically upregulate the expression of VEGF, which is important for vascularization. And coincidentally, BMP2, which is a bone morphogenic protein, and VEGF have been shown to have a synergistic effect on bone mineralization. So we think we're barking down the right pathway. And so we plan to adopt this strategy as we move along to develop our next generation of products. And that is to use existing decellularized allografts that we generate, alter the growth conditions of mesenchymal stem cells to produce custom ECMs, and that would encourage integrin ligand expression of our choice, and would also encourage vascularization. Thirdly, we would incorporate these ECMs, we would transfer them onto the allograft, and then produce these next generation allografts with superior healing properties, which should include better engraftment and retention of stem cells. And those retained cells should be primed for proliferation. And then from the signals already inherent in the allograft, differentiation could proceed for the appropriate tissue, and healing times hopefully would be accelerated. So we are also actively reaching out to anyone who feels that they could branch out a study, a collaborative study on this same kind of thing. And we're both looking for folks who feel that the ECM is where it's at. So please do ask me any questions afterward, and I'll take any questions you have now.